Hello everyone, I'm Pavitra from Excelogen Biotech and today I'm here with the topic ELISA, Enzyme Linked Immunoabsorbent Assay. So what is ELISA? ELISA is an immunological assay commonly used to measure antibodies, antigen, protein, glycoproteins in biological samples. So first described by ELISA was first described by Eva Engvall and Peter Perlman in 1971. So what is the principle of ELISA? ELISA works on the principle that specific antibodies bind the target antigen and detect the presence and quantity of antigen. Moving on to the procedure of ELISA, the first step is an antigen is attached to polystyrene plate. What is a polystyrene plate? Which is a solid surface and is attracted or has an affinity towards bacteria and other antibodies and hormones. Second step is a microtiter coated with antigen is filled with this antigen antibody mixture, after which free antibody are removed by washing. And the third step is a second antibody specific to the primary antibody is added, which is usually conjugated with an enzyme, after which free enzyme linked secondary antibody are removed by washing the plate. And finally the substrate is added. The substrate is converted by the enzyme to form a colored product which can be measured by spectrophotometry. So that is the procedure of ELISA. That is how ELISA works. Now moving on to the types of ELISA. The common types of ELISA are direct ELISA, sandwich ELISA, competitive ELISA and reverse ELISA. So these categories are on the basis of how many types of antigen and antibody are used and how they act. Application of ELISA. ELISA play a major role in detecting a particular disease, and most of the disease which uses the technique ELISA for detection are life threatening. Some of them are Ebola, pernicious anemia, AIDS, rotavirus, Lyme disease, syphilis, toxoplasmosis, Zika virus, carcinoma of epithelial cells. So is that all? Only detection of particular diseases can be done using ELISA? No. ELISA can also be used to determine the presence of antibody and antigen. That is, detection of antibody in food allergens and to determine the concentration of serum antibodies. So these are some of the application which uses ELISA. Not only that, ELISA can also be used for the study of particular substances. Molecular study, immunological studies also uses ELISA technology. So that's all about enzyme linked immunoabsorbent assay. Thank you.